Welcome to Searching for the Question Live. My name is David Orban, and I am very glad to have all of you here following the show. We are streaming. I almost said broadcasting, but that is very much of a television terminology. Simultaneously on Facebook, YouTube, and Twitter. This means that you can send your questions, comments, and if they are relevant and meaningful, I will be able to share them with our guests and answer the questions and make some remarks on the comments. You can also, of course, subscribe to the YouTube channel in order to be alerted when we are going to be live next. And you can also suggest guests. The topics are varied: science, technology, society, and you can also vote on guests that have been suggested by others. We have a Discord community where you are welcome to join in order to follow the conversation, get more in depth. It is also where we collect questions for the periodic Ask Me Anything sessions that I also hold. And finally, if you find this content that I create together with my team valuable, you're welcome to become a member on Patreon at the various tiers that are available: fan, supporter, sponsor, or benefactor. Today's topic is big data. We have grown accustomed to the power of our personal computers, gigabytes and terabytes of storage, and a lot of memory. However, there are just data sets that are too big for a single computer. They must be handled in data centers using platforms that are specifically designed in order to be able to handle these data sets. And of course, they can be in many different areas. Information from retail store loyalty cards, employee databases at large corporations, social media posts and comments, and photos that people upload, all kinds of different sources. One very important area of application for big data is in biology, biomedicine, and this is what we are going to discuss today, illustrating also with specific examples the topic after a brief introduction on the concept of modern molecular biology with Ivo and Murat, who are joining us from Bellinzona in Switzerland, near Lugano. So Ivo and Murat, welcome to Searching for the Question Live.
Thank you.
Hello.
Hey.
Thank you.
So our viewers are from all over the world, and one thing that I like to show them all the time is where I am, which happens to be in Bergamo, and they are, you know, from New Zealand like yesterday, or from San Francisco, or wherever else. And then we go literally around the world in order to reach the location as I show it here on Google Earth, which is a fun platform. Itself a big data platform, of course, because the tiles of all the information that are pulled in real time are amazingly well organized in order to be able to do something like this, and the performance of how the data is correlated and served over the internet is just amazing. And so let me show where Bellinzona is, and the jump is going to be pretty small with a lot of mountains in the middle. But this is where you guys are, in a wonderful valley in the Alps. How did you end up there? Because I am not too politically incorrect if I remark on the fact that neither of you seem very Swiss.
Okay. Yeah, for me, I came here in 2000 for a postdoc at a machine learning AI institute. And yeah, for some reason or no reason, I stayed. And I found a job in 2004 in bioinformatics. And yeah, up till now. Yeah.
And what about you, Murat? Your names are reverse. That's fine. Don't worry about it. But for our viewers, Murat is about to speak.
Okay. I'm originally from Kyrgyzstan, Central Asia, and I did my master's in Turkey. And then for my PhD, I came here, and it was a joint project actually between UZI, that's IDSIA. It's a Swiss artificial lab located in Ticino, Lugano, and then here in Bellinzona, Institute of Oncology Research. So I started my PhD as a joint project. I end up being here, and after finishing my PhD, I'm here for now extra 2 years. Being here is really nice. Bellinzona, Ticino, Switzerland actually has a very nice landscape, weather, nature. Yeah, it's all amazing.
Very expensive, though.
Well, some things are cheap, like chocolate or something.
Chocolate is cheap, and cuckoo clocks or whatever, cheese. And Ivo, you didn't say originally before 2000 where you were?
Well, I've been a little bit everywhere. I grew up in Holland. I'm born in Indonesia. I did a master in Japan and my PhD in London. So I've been a bit around.
Okay, wonderful. And you mentioned the AI Institute. Is that IDSIA?
Yeah.
Okay. So I don't know if he's still the director or whatever his role was, but something like 35 years ago, I used to work with Luca Gambardella.
Oh,
yes, he is one of my PhD co-supervisors.
Wonderful. So next time you see him, tell him that David says hello.
Yeah, and also Jürgen Schmidhuber, maybe you know him. Yeah, he's also there.
Oh, yeah, of course.
Yeah, he's also there.
I have never worked with Jürgen, but we were often at the conferences together one way or another.
Yeah.
So are you either or both originally in AI and then you discovered molecular biology and applied big data approaches to it, or it has always been a little bit of a mix of the two disciplines?
For me, my background is physics, and it was always about simulation and data. When I came, I was in medical physics. I studied like computer tomography of the brain, more the mathematics behind. A certain time after my PhD, I was interested in intelligence to understand it, and I was in this Bayesian statistics, and that was for me a framework to for learning, and therefore I came here as a postdoc to study learning as a mathematical principle. So it was like reinforcement learning and Bayesian learning. From there in 2004, we got a new project about bioinformatics, and I thought in that field there's so much to do, and there's big data going on, and so interesting. So I switched field, but still, it is to me data understanding the data. So it's like what physics people do.
For me, the short answer to your question is yes. I don't have a biological. I didn't have a biological background. My master's was in industrial engineering, where you formulate the problem and try to optimize and find the solutions using the mathematical modeling. And during my master's, I worked on protein structure prediction, and the biology was becoming interesting at that point. And for me, it was like doing a PhD in, since industrial engineering is also a little bit close to finance. So then there was two options for me, like going to finance engineering, or biology-related engineering. And then I found a nice project here in bioinformatics. It was really appealing. I continued with that, and then learned the biology a little bit, and it's really interesting. And then it's again the big data coming, and then most of the techniques that you learn or you know from data science can be still applied, and it's interesting field.
So Ivo, you have been in the field for 20 years. How did the tools evolve? How important it is that the cloud-based architectures enabling you to interface with very powerful hardware available on demand, and the libraries that have been developed, as well as the algorithms, are what they are today compared to the past? Is it just a little bit of a difference, or it represents a very important difference in your productivity, in your ability to turn abstract thinking into concrete use cases?
No, the field has developed tremendously in the tools. I mean, when we started, or when I started in 2004, the tools were, and the algorithms and the methods and the understanding how to process the data was still very early. Also, the computers were still 15 years ago not as powerful as now. But more than the computers, I think, the software evolved and people made more efficient methods, algorithms. And one of the tools that are in bioinformatics very common is R. Is a programming language, R. Just R is a short name, but and with that the bioconductor packages, and there's a tremendous amount of people that contribute to these packages, and the newest algorithms are very fast available in this R bioconductor. Yes, so it became. Thanks to this, actually, the revolution of bioinformatics became actually much easier.
So today, since we are talking about a topic that is important, but also a little bit complex and not necessarily close to many of our viewers, you prepared a few slides, and I told you that if you can go through them in 20 minutes, that's fine. So why don't we try, and then we will see how it goes. I will leaf through the slides myself, so you don't have to worry about it, and hopefully I can follow you as you speak. So please go ahead, Ivo and Murat. Here you go.
Yes. Okay. So what is now happening since 2000 or in this century is that genomics is transforming biomedicine, and through these technologies, the amount of data is growing, and it will promise this personalized medicine. The amount of data is amazing. I mean, one human has about 27 trillion cells, and one cell has 3.2 billion base pairs, and this can be measured nowadays, and it brings like gigabytes of data. So how how can we transform this data to treatments? So you have to go through a low level to a high level. So we have the genomics data, epigenomics, transcriptomics, all this omics data. And finally, we can predict the outcome of health of disease. But data is not enough. You need data by itself is just the raw oil. You have to refine it. Data means information, but if you connect this information is knowledge. Then you need also insight and wisdom to get useful insight. So yeah, the talk was more introduction to genomics. How do we measure it, and how do we use it, right? So
yeah, cancer is a genetic disease. Yeah.
And in 1953, Watson and Crick, they discovered the double-stranded structure of the DNA.
Yeah.
But at that time, we could not really measure it. So the DNA is really amazing. It's in every cell there's DNA that tells the cell how to behave, and the cells in a cell behave all together as an organism. But understanding the DNA might be the key of understanding life by itself. So the DNA is only giving a recipe, but it needs to be transcribed into proteins through the RNA. So if you measure the RNA, you also kind of know the amount of proteins or a snapshot of your. So the DNA is only made of 4 letters. It's A, T, C, G. That's amazing. And actually, your DNA is 90% of your DNA is just junk, and only 10% has some relevant information for making proteins. But the problem becomes that some errors can occur in your DNA. Well, there are some natural variations. Therefore, people are a little bit different. But there are also errors that can lead to diseases, so such as cancer or other disease. So these are parts of the DNA that are deleted or inserted, and these DNA errors, we can now measure. That's the important thing. Here, you see, for example, that certain parts of the chromosome can just switch to another chromosome, and then you get these fusion genes that actually never existed, but they are like a chimera. For certain cancer, they have seen these chimeras. The more older you get, the more you cannot repair these damages, and therefore the cancer occurs more in older people. The mutations, they accumulate. The tumors, they are therefore sick cells that have errors in the DNA, and they find a way to evade death and live forever. And people have understood this by research, and so the technologies that gave rise to being able to measure the DNA. This Craig Venter is the guy, let's say, the Bill Gates of genomics, that did not invented it, but made it very popular and accessible. So one of the first companies is Affymetrix that made a kind of chip to measure your DNA. So they made it actually with the technology of computer chips. They make a similar technology, not the same. They made these chips that could measure your DNA, and this was a revolution. It was around 2000. After that, in 2000, after that, there was next generation sequencing that that has a different technology, but yeah, in the end, could also measure your DNA or RNA in a accessible way. This mining of the, so you measure it, but you still have to understand what the DNA is doing. So that's the genotype. So you have the genotype and phenotype. So the genotype is your DNA that in the end will determine how you look like, the phenotype. But there's a whole process in between of making proteins and how these proteins
effect and eventually bring this phenotype, for instance, disease or healthy. It's not known, and the most important part of the research kind of understand this functional space.
So yeah, if you understand this functional space, you can also interact with it and find, for example, personalized drugs that work for a certain person but not for another person. So nowadays, the treatment for sick people are mostly just one sickness, one treatment. But we have to go to a kind of personalized medicine where each treatment is personalized for a person. So in the end, the genomic data are just huge spreadsheets, but not the simple one that you see, but mostly there are like 10,000 or millions rows and 200 or 1,000 columns. So it's not easy to see patterns in it. So yeah, here you see, for example, the DNA of a normal person. It's a flat line, but in a tumor there are gaps, and there are multiplications of chromosomes that eventually lead to this cancer. So you will see, if you have a cohort of patients, you will see patterns. Here, for example, in chromosome 3, the red, in the third column when there's many reds, there's a gain on amplification of chromosome 3 for this type of cancer. It's marginal zone lymphoma. So if you understand, you see here, you see more summary of it. You see that there's a gain there, and this can drive the cancer, right, to grow. Here we see we have annotated these peaks with cancer genes, and now we know much more how cancer evolves or actually starts. So we can find drugs to inhibit, for example, these driver genes. Here are some methods with networks to model the cancers and their communication of the genes, let's say. This is
it's a part of my PhD thesis. Price collecting Steiner tree is identifying a sub-network in a lot. Okay, all this gene interaction can be designed as a gene interaction network, and then within this network, with price collecting Steiner tree, you can identify subunits. It's the most informative part of the network, like linking the important genes through the most reliable connections to the other genes.
Interestingly, this algorithm was developed, for example, for railway networks. How to make the most efficient railway network through a country. But we can apply the same algorithm for genes. So one of the big challenges is to integrate the different omics data types, genomics with proteomics and metabolomics, for example. And this gives us much more insight, could give us much more insight. The other deep learning net algorithms we can hopefully use in the future or now already if we have enough data. So what we will go, what our final aim is, what I said before, the personalized medicine. So can we predict the drug response from our genomics?
Okay. So, and then another goal is actually to, can we make the analysis available for other researchers, other R&D people as well? Currently, what we observed was the genomics data becoming more commodity. It's more and more available, but the analysis is the problem. And the current way of doing analysis is after getting the data, bioinformaticians are helping biologists to get the an information. However, it's actually, there is a wall between bioinformatician and biologists because due to a lack of technical background, they don't sometimes understand each other, or due to misunderstandings. And then this oftentimes creates lags and takes a lot of time to make sense of data, to make discovery in healthcare. And what we want is to provide self-service analytics platforms for biologists that they can easily upload their data and get the analysis done. Actually, biologists, the other way could be the biologists can do the analysis, but in order to do that, they have to learn statistics and coding, which is oftentimes it's very frustrating. It takes long. So biologists actually it's hard for them to learn coding. I mean, we have been seeing that in the lab. Very few biologists actually can take care or can pick up and learn quickly. So, and there is always gap between biologists and their data. They're much dependent on bioinformaticians. And with such platform, biologists can easily interact with their data. So that's the whole idea behind. And converting it into a self-service platforms, like very similar to the self-service checkout in the markets at the moment.
Yeah, self-service is fast and
convenient,
cheap, and you can do it yourself, right? So that's our analogy.
Wonderful, wonderful, very, very interesting. So I know that you also have a specific example that you want to show, which I think will be very informative and important. Before we do that, I wanted to share a question that comes from Eyal Fischler. He says, I have a feeling that saying that DNA is 90% junk would be like saying 640K RAM would be enough. So I don't know if that analogy stands at all, but what he means probably is that maybe it is only our contingent understanding of the nature of the DNA that makes us say that the redundant part serves no purpose. We pretend to be smarter than 4 billion years of evolution that decided that it was better to keep all that baggage because evolution tends to discard what is not needed and only keep what is necessary. So I don't know what is your thought about that.
No, it's definitely we at the moment think it's junk, but maybe in 20 years or 10, 20 years, we know what it means. Now, certain people think already that maybe it's not junk. Maybe they are like a sponge for attracting other RNA, DNA. So, yeah, our understanding at the moment is just not enough, or thinks it's just, yeah.
Another remark that I think could be useful is that in lay terms, when we talk about cancer and tumors, what we say is, oh, lung cancer or cancer of the liver or this or that, imagining one cancer per organ without realizing that almost every cancer is unique. there are families, but already the families are dozens or hundreds of families. And then, of course, how it happens in each individual is also very different. That is why, even though we have been trying for the past 60 years, we haven't been able to develop a single shot cancer cure, except for rare exceptions in those cancers that have been recognized as being actually caused by viruses. And then we developed vaccines against those viruses so that the infection doesn't happen and the cancer doesn't develop, like it has been done over the course of the past 10 or 20 years for the papilloma virus that was found to cause a cancer of the uterus. So there are those people who think that the vast majority of cancers are going to be ultimately recognized as being caused by viruses, and we will see that is the case or not. But in the meantime, personalized medicine is necessary exactly because a single solution is not applicable to everybody when we get cancer, right?
Yes, nicely outlined. That's right.
Yes, the mutations that you accumulate is per person different, and you don't have only a few mutations, but you accumulate hundreds of mutations. But at a certain time, you have these driver mutations that are shared with other people, and eventually.
And also, of course, the role of the immune system in all of this is fundamental. When we are young and we have a very, very active immune system, even though we have literally hundreds of cancerous cells in our bodies at any time, none of them have the chance of developing into a tumor because the immune system goes and attacks them. But as we get older, our immune system is less active and less able to catch the cancerous cells, and they have the chance of developing into a tumor.
Yeah, but many cancer cells also develop a strategy to hide themselves from the immune system.
It is an extreme example of anthropomorphizing, of course, but I heard you say, I don't know if Ivo or Murat, saying that the cancer cells are sick cells. If I were a cancer cell, that would sound offensive. I am sure I would think of myself as a perfectly fine.
That's a perfectly fine cell, right?
I would be very proud. Actually, I would be convinced that I'm much better than all the others because I'm immortal.
Because
I would say I'm fantastic, and all the world
is seeing it.
Yeah. Well, I don't know if we are going to be able to find a truce with cancer cells to say, listen, I let you be as long as you don't kill me. That could be another therapeutic path that we may explore in the future. So let me share your screen, and you will be in control in order to show us your platform. So what we are seeing is a specific example applied to the COVID-19 data set of your startup, which is called BigOmics, and your platform, which enables non-data scientists to play with data. Is that correct?
Yes, that's correct. This version, we call it Viromics because we collected previous coronaviruses, SARS, MERS, as well as some of the current SARS-CoV-2 coronavirus infectious data from public databases. It's around 200 now data sets already injected, and this is if user can, it's already running on covid19.bigomics.ch. User can read the manual, watch some tutorials about how to use the platform or Docker image, and then hit the play. This is the landing page. And for biologists, if they would like to upload their own data, they can come to upload data and see example, bring their data set similar to this example and upload it. This is the visitor map where we see like in last quarter, more than 5,000 people used it. One interesting thing is it's very similar to what's going on in terms of pandemic in the world now. It was very high in China first, in the beginning, and then Europe. Now we see that US is really high. So it's interesting. And then they also can pass here their questions if they have. On the other hand, as I said, there are 200 already virus-related data sets. This is a recent proteomics data. User can load it. It's pre-computed. It needs some seconds to load it, and then it's gonna be, the data is gonna be ready for visual analytics. Usually, biologists would like to start from this step. What we call it clustering. It's a holistic view of the data. This is a heat map that biologists really like it, where samples are on the columns and genes are on the rows. We see like, okay, here this red is those are the genes that are really high active in these virus, you see. And then it's really interactive. People can find all information about the methods, what we have used it. They also can play with the settings and set up it. They also can download as PDF or PNG for their publications or presentations. Another very interesting figure for biologists is this PCA or TSNE. Again, it's interactive. This is another parallel plot, what we call, just to give an idea what it can give is, okay, this 4 is a beginning control. It's a normal, healthy patient, healthy individuals. And then the virus is the diseased individuals after virus enters the body after 2 hours, 6 hours, or 24 hours. And then for biologists, it's one interesting thing is to get the pattern as quickly as possible. Here they can see that this group of genes are really deactivated or less active when virus enters the body, and then they can immediately see what kind of biological processes associated with that collection of genes. Vice versa, they can also see what kind of biological pathways are getting really active. And going deeper in terms of analysis, one example or one thing that biologists like is the comparing, again, the diseased versus the control, again, 24 hours, and see which genes are upregulated in one group or the other. And then they can get it. And the one good side of our method is, our platform is, we provide multiple methods where it's good to have a consensus, and they can see how many methods found that were really active for that specific gene. Another interesting thing I think would be they can easily associate their own data to the public under this signature, and then find similar experiments, where for their experiment they will gonna get what are the other studies that are showing similar pattern, and specifically which genes are the similar or non-similar. You can easily see that.
That is also visualized in this 3D plot. So you can visualize the experiments as a dot in 3D space, and you can see which one are. So you have a cluster of experiments here in bright red that are very similar to your, let's say, data.
Another interesting thing is gonna be this drug CMAP, what we call
drug connectivity.
Yeah, drug connectivity, where it's for a given comparison. We collected around 5,000 drug profiles and then correlate for given profile and identify the, for instance, in this case, it is, and then identify the drugs that can reverse the effect of the virus. And then we see that fostamatinib is in the literature is also referred as one of the possible treatments for the virus. Okay. All in all, it's very interactive. For biologists, they don't have to really code. They have to just come and then upload their data and get all this interactively, all the plots. For instance, if without this platform, usually if they would like to change some parameters, biologists have to ask bioinformaticians, and then this plot has to be from scratch, has to be created.
Yeah, what I said before, there are bioconductor packages or tools. They were not really made for the biologists. They were made for the informaticians and the bioinformaticians that can program. So we are now in a stage that it's not the right tool anymore for this kind of.
So this is absolutely fascinating and really powerful, at least for those people who understand the visualizations, because of course,
and those are the biologists.
You don't eliminate everything, but if you are a biologist, evidently it is extremely helpful. And of course, your platform allows any kind of data set to be uploaded and analyzed similarly. This was just a specific example of a large number of data sets around COVID-19.
Yes, it's mainly, this platform is for transcriptomics and proteomics data sets.
And what is the universe of your market? Are there 100 people in all over the world that would be potentially interested, or 100 million? What is your target market?
So what we can say is the target market from top down, from the information, it's expected that by 2025, the market will be 41 billion. And that's the, in terms of the, let's say, data generation, we know that it's exponentially growing. And by 2025, the data actually generated by genomics will overtake all social media, including YouTube, Twitter. So it's exponentially growing. The market is huge, potentials are huge, and it's currently mainly in healthcare, but we already observed that genomics is entering in food tech and agriculture. It's another big markets. And
I can also see other markets like sports, for example. You know, to understand what kind of training regime a given athlete needs that is different from what he or she would receive just based on the experience of the trainer, as well as many others. Exactly as you said at the beginning, a lot of application areas depend on the ability of the tool to be used in terms of how easy it is on one hand, and on other hand, how expensive it is. So does it cost millions of euros to use your platform?
No, exactly. It's not millions of euros. What we offer is a paid subscription. It's very affordable for individual biologists as well. Currently, having a bioinformatician might be very expensive, and not all labs, biological labs, I mean, or groups, can have bioinformatician in-house. So our platform is on the range of a couple of hundreds per month, I would say. And then it's for individual cases. And for enterprises, of course, we have more advanced analytics, on-prem installation, as well as customized solutions. And the price difference is it's different.
Yes, but we also have a free version, actually, open source for academic individuals. So
this platform actually is now open access, what we showed in Biomedics. So
yes,
yeah,
I think the interaction of what is openly available, which is close to the heart of a scientist, and obviously anybody researching these topics wants the broadest accessibility and availability. And on the other hand, how and when the commercial opportunities are safeguarded either via patenting or industrial secrets, so that the economic sustainability of a given solution is preserved. So your platform allows both open and not open approaches, depending on the project or the needs of your client?
Yeah, well, we have open version of the platform, and then we also have some closed versions that's for enterprises. So, and then, for instance, free versus paid subscription. There, in free, still biologists can upload and analyze, but with limited analysis, limited number of samples, and they cannot save their data on cloud. On the other hand, on paid version, they can really save their data, access to higher number of public data sets, and the storage is another convenience for them. They don't have to really compute every time when they visit our platform.
Yeah. So the idea of open source is always like, okay, you can see my source code, it's transparent, it's free to try.
Yeah.
But we build a service around it. So this is, in our case, the cloud service where you can access it everywhere. Your data is stored, and you get support of how to use it in case of troubles. So yeah, we are very, very fond of open source. We grew up with it, so yeah, we decided, no, this is the way to go.
Now, you showed a map of the visitors. How do people find your platform? Are you ranked high in Google for certain keywords, or they hear about it at conferences? How do people find out that it is available?
You mostly answered your question inside your question. It's yes, from our website, from these keywords. We also try to go to conferences. But on top of that, we mainly advertise it on our social media, through LinkedIn, through Twitter, where we explain our platforms, keep the updates and all other developments related to our platforms, and announce it on our social media.
Since you operate at the intersection of two very important but also very specialized fields, do you believe that at a certain point as you grow, it is going to be hard to find talent to add to your team?
Well, I mean, finding a talent.
Well, part of the idea was this. I mean, yeah. You're right. In the intersection, there are certain experts with bioinformaticians, and with the growth of the data, there will not be enough, and it's hard to find these experts. Therefore, our idea is to make a tool to invite the biologists to do their own data analysis, and that makes the pool of people that can handle and understand the data much bigger, which will be necessary, and it's already kind of necessary.
So you, as BigOmics, the startup, you will hire your best users. Your users will be your talent pool.
And the other idea was making open source, for instance, showing the code and then check who can contribute, who contributes the best, and then we can select from there.
Unfortunately, we cannot. We need also users that can program.
Yes.
So it's not just the biologists. end user, but really for our purpose, for making the tool, you do need good programming skills. And if there are such people, please contact us.
Of course, absolutely, absolutely. You are also fundraising, if I'm not mistaken. So if any investors are watching this, they should also be excited about being able and jump on the wagon of an exploding set of industries like big data, artificial intelligence, biology, bioinformatics, health. All of these combined in BigOmics. So Ivo and Murat, thank you very much for being with us. I will now close our stream and say goodbye to our viewers, but don't leave. So thank you for the presentation and all the information that you shared.
Yeah, it was a pleasure.
Thank you.
Really,
it was a pleasure.
Yeah.
So thank you, everybody, for being here with us with Searching for the Question Live, for your questions and comments. If you speak Italian, you can also subscribe to my Italian channel on davidorban.com/YouTube Italiano. And you can also do the same, suggest guests, vote on guests, and so on. And as I mentioned also at the beginning, if you believe that this content is interesting and valuable and you want to see it keep going, feel free to become a fan, a supporter, a sponsor, or a benefactor on patreon.com/davidorban. So thank you very much. See you at the next episode of Searching for the Question Live.